Welcome to the Proteomics in Proximity podcast, where your co-hosts Cindy Lawley and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Cindy and Sarantis.
Hey everyone. Hello and welcome back to Proteomics in Proximity. Thanks to our 11 listeners at Sam, Ray, Carolina, others. We're grateful for your attention and your feedback. And our listeners have given us some valuable feedback over time, and they've found us through different social media avenues. But to make that easier, we're announcing that we've got actually an email address now. So we'll put this into the show notes, but it's just PIP for Proteomics in Proximity at olink.com. And we'd be happy to hear from you around suggestions you have or any interview recommendations you might have. And with that, today we are talking to Evan Mills. Evan, I'll let him introduce himself, but he is an illustrious pharma executive here actually at Olink. And we're excited to talk to him about how pharma are finding proteomics super relevant on many different levels. So with that, let's get on with it. Hey, Sarantis, how are you?
Hello, I'm fine. Thank you, Cindy. Welcome, Evan. Looking forward to hear from you. All the great news.
Likewise. Good afternoon, Sarantis. Good early morning to you, Cindy, on the West Coast.
A little dark over here.
Little dark, but that's all right. No, I'm really honored to be here. And I've been wanting to talk about how proteomics and the pharmaceutical industry are aligning for really exciting things. So very happy to be here.
Can you give us just a little background on your history in this area? You've been in this for a while.
I have. So I was a bench scientist, really, passionate about oncology and neuroscience research. I did some work at Yale University for a while. And then I got into the sales commercial side of this world, started actually in the pharmaceutical sales industry, which was exciting because of the opportunity to help patients, right? But my real passion was in the science. And about a decade ago, there was a very innovative proteomics company that caught my attention. And that's where I started this journey where I've now been at Olink for over 5 years. And supporting the most innovative, ambitious researchers in this multi-omics space has just been a phenomenal journey. So my background is I loved science. I want to help people. I want to have some sort of translational impact with the work I do. And right now at this moment, there's never been more momentum in that direction. It's really, really exciting.
It's very exciting. We just had here at Olink 3 pretty exciting Nature papers come out in I think it's the online October 4th, but the print journal October 11th with a beautiful frog on the cover. It's it's an exciting time with those 3 papers. So those include a lot of applications around why pharma would invest in proteomics. So I'd love to get your thoughts on why did 13 pharma come together, invest in proteomics? What's the outcome? What's the result that they see out of it?
Yeah, I mean, that's been a real career highlight, is being able to be involved in that project from its inception. And, Cindy, with your background in genetics, right, there was a previously formed consortium around whole exome sequencing in the UK Biobank, and then eventually whole genome sequencing. But there was this idea, and I was having lunch outside of a Harvard symposium with Dr. Chris Whalen, very smart geneticist who was at Biogen at the time. He's now at Janssen. And he just asked a question. He's like, hey, we're thinking about what makes sense to do next. We have all this richness in the genomic data, but we want to do something closer to phenotype. Would it even be conceivable for Olink to run 50,000 samples? And this was before, some of the innovation that would have made that possible. And we said, yeah, I think we can do that. I think we can get there. I think we can do that. So it was just born out of curiosity and the desire get closer to phenotype. So the goal really of this ambitious project was, can we both better understand drug targets that have causal links to disease, and can we simultaneously find biomarkers to help the drug development process? Because obviously with proteomics, you can do both, right? It can act as a bit of a filter to tell you which of these genomic disease associations have a plausible biological story, and which ones should be pursued and which ones should perhaps be killed. But simultaneously, you can develop a suite of tools to determine risk based on proteomics, to determine disease progression based on proteomics, and to discover biomarkers, which are obviously always desired to aid clinical development. So, I mean, we're just starting to see all the publications. We're starting to understand all the utility that's going to come from this data set. And it was just such an ambitious, smart idea by Chris, and then eventually Melissa and Lyndon and Brad, the countless others who contributed to the project.
Evan, going back to this journey, that's amazing journey. How easy or difficult was to convince the genomics community? Because you mentioned it was like a heavy genomics community, right? Change the mindset in a way that, okay, also proteomics can add value. How easy or difficult was this process?
It's really hard. I think it was really hard because if you just think about the tools one would need to develop to measure, right? Our DNA is very nicely organized into a helical structure. There's 4 bases to measure, and Illumina and others now have developed amazing tools that can measure that at scale. Think about the proteome, right? There's 20 amino acids. They combine in a myriad of different ways. I mean, it just is such a formidable challenge that geneticists would say, I'm not so sure, right? I'm not so sure the tools exist. And, oh, by the way, we can measure everything with genomics. We can measure everything. And you're approaching us with something that measures 1,500 at the time, right? And then 3,000 of what people assume would be maybe 20,000 proteins that you could try to capture in plasma serum. I mean, that there's a big debate about that. So it was challenging, but the obvious central dogma of being closer to disease and things that are reflective of real-time biology versus your blueprint of your biology was compelling enough for them to give it a shot, but it was not easy.
So you must have focused on what is the near-term return on investment for pharma for running a proteomics project. And I would consider this UK Biobank sort of PQTL, developing therapeutic targets, all of those things you've already mentioned as more mid or long-term goals. How did you, I think it's a great question Sarantis asked, how do you talk to them about what you believe is the value? And I will also say Gary and I looked, Gary is our illustrious person who manages our database of over 1,400 peer-reviewed publications. And he has seen over 84 of those are pharma relevant publications. So there's a significant number of publications that have been put out there that document some value to pharma, but that's pretty recent. How did you approach them when you first came to Olink?
Yeah, no, it's a good question. So we can take a bit of a sidebar from the UK Biobank discussion, because really, fundamentally, drug developers are trying to bring effective therapeutics to market faster. And they also invest enormous resources into each program. And it takes, 10 to 15 years on average, to get something approved.
And how many millions of dollars, right? I mean, it's just, yeah.
And patients.
And then what, 90% of clinical trials fail, I think, or somewhere around there?
Yeah. I recently had a discussion with an executive vice president of research at a major company who said he would be the world's best drug developer if he failed 80%
Isn't
of
that
the
wild?
Like, if he could go from failing 90% of the time, he would be the best. And it's right. It's just such a high attrition game. But no, so Cindy, the way that a lot of people in the industry are starting to look at this is with population scale proteomics or high throughput proteomics, you can learn a lot about things you've already invested in. So let's say that you have a drug that's approved, such as can never say that correctly. Jardians. Let's go with Jardians.
Yeah.
A very, very effective SGLT2 inhibitor used for the treatment of diabetic control. They've also noticed after having it in enough humans in the wild that there's significant benefits to heart failure. So if you can access, and this is one of the publications that you referenced, if you can access samples, right, from completed clinical trials, and most companies are sitting on these, they're just in their freezers, waiting to be analyzed if they have the exploratory consent. If you take a look at proteomics at scale from lots of humans treated in the clinical setting, you can learn a tremendous amount about why certain people respond and certain people don't, right? That's the holy grail essentially, is can you proactively know which patients should go on which therapy?
And we often call that
You can
stratifying
understand-
patients, just to use the term that we've used before.
Yes, absolutely. And, understanding the mechanism of these drugs, right? Because, you have a target, you have a hypothesis, you test it in cell-based models, animal-based models, but you don't really have a chance to look at scale in a human population to see how it impacts the human body. So then with that data, you can better understand why there's this benefit in an indication for which the drug was not initially approved. You can understand, what other pathways are being impacted by your therapeutic? Are there repurposing opportunities? Is there a way to very rapidly take this thing you've invested in, this asset, and figure out that there's more places that you could help people? There's more indications where this drug would actually be a really good fit. So that's a very short-term win. And we've noticed multiple clients building this as a strategy to take Olink proteomics in this case, to better understand already approved drugs, which in some ways is counterintuitive, right? I mean, ideally you'd think from the beginning, you would want to know everything you can about the drug. But there's this reverse translation movement that seems to be bearing quite a bit of fruit for the industry.
That was actually my next. That's my next. Sorry. That's actually my intriguing. This is my next question, Emma. Do you see now this trend of a strategy in the pharma big pharma company, because you talk with executives, right? And you know the strategy and you discuss about this. Do you see this coming? Do you see that using like proteomics, a big number of data to reposition the drug, for example, to identify mechanisms of action, even the late stage?
And why would
How is your feeling?
And why would they ever publish this, right? We think of pharma as needing to hold these things tight. So great question, Sarantis.
That's a good question. So the answer is yes, in pockets. I think it's just becoming a strategy for the more innovative companies, right? There's always some concern, right? For ongoing trials, do we really want to know that much at a phase 3, right? If we have a candidate compound, do we want to do exploratory research? Maybe we find something we can't explain. Maybe we find some safety signals. So what I'm describing is drugs approved. Let's extract as much value from that asset as we can. And there's definitely companies that are taking that on as a strategy. And to your point, I mean, having gotten to know folks in pharma really well for the last decade, I mean, they're great scientists. I think I mean, not to insult any of my academic colleagues or people I've worked with or people that, I've supported over the last 20 years. I think there's incredibly talented scientists that see the opportunity to have a fast path to impact. And they do want to share. They want to publish. I mean, look at this consortium. It was 13 companies that are competitors coming together.
I was complimenting one of the pharma researchers on a new hire from academia. And she was saying they came to me because they're a physician, an MD, PhD. And they said, I can help one patient at a time in my practice. But if I come here and do more broad-based research, I can affect millions. And I was like, wow, that's an interesting perspective. I like that. And it lines up with what you're saying. I think a great example of reverse translation that you've talked, I think one of the examples you've talked about in the past of this, taking samples that are sitting in the freezer where a massive investment has been made is the one from Samina Ticau, and Paul Newey. Paul, of course, is also on the UK Biobank flagship paper that came out last week, this week, whichever onliner you want to reference. Can you tell us about that example?
Yeah, this is a really, really interesting story. And that this originated about 5 years ago and was published in 2019. So it's a bit dated, but I think that the point is incredibly powerful. So, hereditary Transthyretin-Mediated amyloidosis is a genetically defined
and can
disease
I just say,
that
you
really
can pronounce
has
that, but Empagliflozin is pretty darn hard. Yes. Sorry. It just seems funny. Jardians. Anyway, no, back to HATTR. Sorry.
HATTR. No, I've probably told that story more times than
Jardians. Exactly. Oh my gosh, it's hard.
So HATTR is a really, really debilitating disease with a variable rate of onset. So if the hereditary form, if it runs in your family, right, you can be screened to know if you're a carrier and know if you're at risk for developing the disease. Alnylam developed a drug, patisiran, that is an RNAi-based therapeutic, where they are very effective at slowing the symptoms and helping these patients. However, even with this genetically defined population, it was hard to know when the disease was becoming active, when these patients were a good candidate for treatment. So they ran a retrospective study. This was before Olink had an NGS readout, so it only measured like 1,100 proteins. And they discovered neurofilament light, which is a very ubiquitous biomarker for, neuronal damage. But they found that this biomarker, neurofilament light, was indicative of disease progression, was also a biomarker of efficacy. So after patients were treated with patisiran, it dropped significantly. And it was a disease biomarker. It was fourfold elevated in the patients versus healthy controls that they measured in the study. And so now what's really interesting is there's a protein-based assay that could give treatment decision information, right? So it's being validated, and it's only a single biomarker, and it's a ubiquitous biomarker. But in this subset, proteomics is giving you some actionable insights in a genetically defined population where they're now developing cutoffs to try to see, hey, if you come to your clinician and NFL is measured, and once you hit a certain cutoff, that might actually indicate, even though you don't have symptoms, the disease process has started and you are a candidate for treatment. So it's great for the patient. It's obviously great for Alnylam, so they can, justify patients getting on their therapy. And it was where a proteomic screen, right? They didn't know what to look for. They didn't have this hypothesis. They just wanted to see what's changing in these patients after treatment, what's changing over time. And I think that's a powerful way that unbiased proteomics can point us in the direction of actionable biomarkers to help patients and clinical development. So yeah, that was a really interesting story.
Great. Actually, I would like to go a little bit back because you mentioned about pharma and academy. And then we know at the beginning it was really difficult to communicate, right? The two worlds. They were like separated, academic research versus pharma research. Do you see this changing? And would you see a benefit of this change?
Yeah, absolutely. So we just got off the phone. Cindy and I were just on a call with a really, really impressive academic researcher who mentioned that she's on the board for two very large, important studies that are being run by pharma companies, right? She's an expert in her field, and she's advising on how they should spend, their research dollars to best move, very important therapies through the clinic. Yeah. I see it happening all the time. I mean, so our team focuses on primarily pharma and large population cohorts, right? And there's incredible connections between the two, right? Because if you think about it, if I'm a pharma company and I'm interested in atopic dermatitis, for example, it would behoove me to really profile with all these new omics technologies as many patients from the best cohorts in the world that have atopic dermatitis. You could do that through a population cohort. and there's going to be some subset. What's probably more efficient is to work with KOLs in the field.
Yeah.
And then they've collected the samples.
Yeah.
You provide the resources. And then with that, right, if from the protein side, you could discover, are there disease progression biomarkers? Are there endotypes? Are there subphenotypes where there's slightly different molecular drivers that we could then approach with different molecular, entities that we either have or that we could develop to have, a higher rate of success in the clinic.
Precision medicine.
So absolutely.
No.
Yeah. And that's been a term that's been really kind of reserved for oncology, right? Primarily.
Yeah, but
And I think that's because the tools have existed at the genetic level, and obviously cancer is a very genetically driven disease. But if you look at, some of the more multi-system diseases that, in the cardiometabolic space, in the autoimmune space, proteins, I think, will be the next big thing in terms of finding signals that can differentiate subtypes of patients and then give them better treatment options in the future.
Would you talk about cardiometabolic, would you consider like a blockbuster kind of disease? Would you see now pharma investing more on this or expanding on this research? Because for me, it's the impression that we are moving a little bit far away without, of course, leaving behind the traditional type, if you can say, disease like cancer. I see that now pharmas, they are going to rare disease, they are going to cardiometabolic disease, they are going to obesity like a disease. What is your feeling? What is the blockbuster in the upcoming years in the pharma?
I mean, without getting too philosophical about why, the GLP-1, GIP-1, all the Lilly and Novo competition and others, and there's Pfizer and a lot of other companies are getting involved, right? There's just a huge societal issue with obesity, and there's enormous amounts of investment happening in that field. I do think that there's a bit of a gold rush right now, but scientifically, what's really interesting is, it's not just about obesity. I've been fortunate enough to talk to some of the leadership at these companies who are really trying to develop the next, Mounjaro, the next semaglutide. And what they're noticing is there's so many knock-on benefits and there's so many, benefits to multimorbidity that they want to both understand at the molecular level, what's driving that, but also understand, are there patients who have a more aggressive form of obesity, for lack of a better term, right? Is there a subtype of patients that really need 30% weight loss or 40% weight loss? So it's a fascinating effort. And I mean, given the reality that it's a very environmentally driven condition, proteomics, I think, will be an indispensable tool. I mean, again, the other day talking to, a KOL in the space saying, these companies and society generally says, well, let's do genomics first, right?
Yeah.
Like we have all these samples, we're going to just do whole genome sequencing and see if there's some sort of signal in the genetics that's going to help us answer these questions. And they're starting to say, hey, wait a second, like there's these proteomic tools now that, don't you think it makes more sense in obesity to look at the proteins and they're dynamic and you can look at multiple time points and see what's changing post-treatment, et cetera, et cetera. So it's just an interesting side note that in this field, I think proteomics is going to be particularly valuable.
And I just want to define a couple of terms. So KOL is key opinion leader. We use that a lot, lingo around here. People that are driving and influencing decisions that are happening out in the field. Particularly, we are thinking in terms of genetics and proteomics. And then the semaglutide and these GLP-1 agonists that Evan mentioned are not only relevant in obesity, but they're actually being almost prescribed where people pay out of pocket in some offerings. So I've met people that are really keen to be on them or are on them and have had a lot of success in reducing their maybe not in the obese category, but an overweight category where, again, you can expect, based on what we've seen, health benefits there as well. So I just wanted to throw that in. Really interesting space, right?
Yeah. And maybe also on that note, a lot of these drugs, a lot of these inhibitors, as you mentioned, Evan, they are influencing more than one disease, right? They are targeting more than one, and I think that's also where pharma would like to know. You have a drug more than one disease. Is that my feeling, or have you seen this happening from your perspective?
Yeah, for sure. I mean, it's then that's where the deeper understanding of the mechanism of these drugs, right? Which, yes, there are great model systems that, and if you using a cyno model, monkey model, eventually mouse models, rat models, there's all kinds of models, and you can get a good sense of how your drug's behaving. But, often with these phase 1 or phase 2 studies, the amount of patients is fairly small. You can get an idea, but that's why I believe that, companies are investing significant resources to look at the bigger studies, right? Because you can just see, you just get more statistical power. You have a better chance of really understanding how my drug's impacting multiple pathways, multiple organ systems. And then once you have that knowledge, it just makes you so much better informed for new therapeutic, ideas, even just repurposing the existing therapeutics. So yes, Sridharan, I mean, there's the more indications, the better, right? I mean, just from a simple, pragmatic business perspective, but having the molecular, justification, I think is what, as a society, we should all ask for, right? I mean, just seems to be what they
especially when we're consenting, and all of us, if we're, participants in these sorts of trials. I think another promise here, and we're going into ASHG soon, and we've got 25 different posters of folks leveraging some proteomics from Olink, which is really exciting to see. This is a genetics conference, and clearly there's this value of layering the genetics onto the proteomics onto the genetics. There's also 7 talks. That doesn't include the talks that we're sponsoring. So I think in this environment, I guess I'm wondering what are you most excited about, Evan? Sorry, I just
No, that's fine. I mean, it's a hard question. Let's just talk about this environment being the fact that there were 3 publications in Nature, right? 3 publications that just dropped about the promise of population proteomics, right? Yeah. So, I mean, I just think it's the beginning, right? So 50,000 samples from a largely Northern European cohort has led to a treasure trove of insights, right? 14,000 associations, 80+% of which were novel. People can dig into that for a long time.
And reference back to it with their own studies to
that's a great point.
Corroborate the signals that they're seeing. I think we've talked about that before. Yeah, go ahead, Evan. Yeah.
And that's super exciting, right? Because that will provide a bit of a backbone to understand causality and give us insights into drug targets and biomarkers. That's great. But it's just a small subset of the world's available resources from a cohort perspective. So there's enormous benefit to going bigger, as the AstraZeneca rare variant paper shows, right? To capture these rare variants. And this is what, Regeneron Genetics Center has done for years, right? They're doing genomics on all of these very large populations, these founder populations, to find these signals that really come out when you go big. That will happen at the protein level as well. I think going to different parts of the world, right? There's just going to be enormous richness as we go from without question. Everyone wants to do that. But if you say the thing I'm most excited about, to be honest, is proteomic risk scores and the potential for a whole suite of tools to help perhaps, consumers one day, certainly drug developers, perhaps health insurance companies. Who knows where this all goes? But, speaking to Ben Sun and some of the head analysts from the UK Biobank project, they, with just 50,000 samples and machine learning and AI-based algorithms, are able to pick up on these patterns, right? Of sometimes a small number of proteins. I believe Claudia Langenberg and Robert Scott had a paper where it was between like 5 and 20 proteins could distinguish your risk of a large number of common diseases. I think once those are validated and those are refined, that is a game changer, because then if I'm a drug developer, I can apply these algorithms to all my clinical trials and better understand, hey, are we on the right track? And what other impacts are we having on a wide range of diseases? I mean, to me, that's incredibly exciting, and it's not without its challenges, right? I mean, you have to validate these things in sufficiently statistically powered studies. But one could imagine that there could be a suite of tools in the future based on, a manageable number of measurements that could be used clinically. And that's where I think the next big evolution will be, is taking this data that's been generated by either academic funding, pharma funding, government funding, to really look at a lot of diseases at the protein level at scale using these new proteomic technologies, and then whittling it down to things that are clinically actionable that you would have never found if you didn't take a broader view, right? I think that's the difference.
And just to double click on those authors. So there's Ryan Dhindsa on this rare variant paper. He's the first author. He's at Baylor, working also with AstraZeneca, where Slave Petrovsky is the PI on that paper. There's the Ben Sun, who you mentioned, and Chris Whelan paper. That's our flagship paper. We consider it sort of the broadest group from the UK Biobank Pharma Proteomics project. And then, of course, I want to also just touch on Grimmer Elvarn and Kauri Stefansson's paper. Well, if only to highlight something Kauri said about proteomics in general, and that was along the lines of what you're describing, that proteomics, that an algorithm they've been able to develop with proteomics can predict all-cause mortality in any individual. So how many years does one have left to live, right? So if I go into a clinical trial and I've got a prediction of 30 years left to live, and then I go on to this drug, and partway through that trial, or maybe 3/4 of the way through that trial, you look at my proteomic score on my prediction, on my how long do I have to live. This is a way to have very short clinical trials that actually are representative of a longer period. I mean, imagine a depression trial. I remember there was one trial on depression. It was something like 6 weeks, right? If you're talking about major depressive disorder, a 6-week window is a hard one to draw conclusions from. And we do the best we can. But having something like this that is a reflection in the future of what this is doing to your proteins, I think is very exciting.
I'm thinking out the era. That's mean, let's say there's the era of proteomics versus big data generation for biomarker discovery, right? Then what is coming next? The in vitro diagnostic era booming, for example. Then some of these biomarkers be like customized and used for clinical diagnosis. How will you see this roadmap? I know it's difficult to predict, but will you see this coming actually from your perspective?
I think so. And Cindy's point, I think, is really, so I sort of touch on that real quick, and then I'll touch on that, Sarantis, because they're certainly connected, but slightly different in my view. So this idea of having a risk score to help, shorten a trial, right? Give you some sort of a surrogate endpoint or some sort of early read. I mean, I remember, Kauri I believe in a presentation he gave, mentioning that, if you could apply this, risk score, you could cut the time of a cardiovascular outcomes trial, significantly, I think by more than half, and save hundreds of millions of dollars, right? And I think broadly that would help everybody, because the companies developing therapeutics would not have to spend so much money. It would be less expensive, and the right patients would get, the right drug, because they're at higher risk if you use an enrichment strategy. So I think that's absolutely coming. There's no doubt about it. But then, the real end game, I think, Sarantis, is what you've referred to in terms of this in vitro diagnostics piece. So I was recently visiting Roche Diagnostics, in Basel, and they're, world leaders in diagnostic tests. And by and large, today, it's a singleplex assay, right? You're measuring one thing.
Yeah.
And there's a lot of reasons for that. It's challenging to have multiplexed assays validated to the level that today we're used to, being required from the FDA and others. But biologically, and just, if you just think about the complexity of disease, a single marker is probably not the best thing to do. So I do think that's coming. And I hope in the rest of my career, I have a role to play in that, because if we can have very predictive multi-marker tests to be used in the diagnostic space, that to me will be the biggest societal benefit that can come from all of the amazing work that's happening right now. I think that that's where this all goes. And you can just imagine a future where there's much more resolution to your personal risk for disease, your personal response to therapies, that we just don't see today. So yeah, I think that's where it goes. It's a hard road.
Well, but we're already seeing multi-gene tests in cancer, right? And stratifying and diagnosing to help better serve cancer patients. So I think, and yet there's still a lot to be done there. And I think, the pan-cancer study that came out of Matthias Uhlen's team, which we've talked about on the podcast before, is a great place where proteomics is making inroads. So yeah, fantastic.
Also, to add also on that, is like, as you said, that one biomarker, it's not enough. And we have a lot of examples in papers in Olink, right, where you saw additive value, prognostic value of having more than one biomarker. And they're really great from Zannad, Matthias Uhlen, there are plenty of papers that show that this additive value. And I think slowly the community start realizing this, that having more than one biomarker increase the value. That's great.
Yeah.
Yeah. And it just depends on who you're talking to in terms of what they think the next big thing is, right?
Yeah.
You asked for my opinion. I gave you my opinion.
Yeah.
So someone else could say, hey, I just want to measure 10, million samples, and then we're going to get much richer insights into the next best drug targets. And then that's going to create more efficient pipelines and a better, drug development universe in the next 50 years. And yes, I think that'll happen too. But there's just on all ends of the drug development spectrum, these innovations, that Olink and others have made, I think are really, really going to be transformative. And they already are, but it's so early.
Yeah.
I'm sorry, not to go on a tangent, but like, it really is early.
Yeah.
And part of, with whole genome sequencing, right? The cost dropping has really enabled things.
Yeah.
That's an important point.
To be honest, it really is. It's if we're just going to be frank and honest about, the opportunities to help as many people as possible. If a tool is prohibitively expensive, it's never going to have broad adoption, And when I joined Olink, things cost a certain amount of money, and now things cost less. Like it's definitely,
and we get more.
Yeah, there's been
we get more from it. Yeah.
Yeah, exactly. There's more data coming out for a lower cost, right?
Mm-hmm.
And that's what the market has expected. That's what people, are demanding. And again, that's very hard. It takes a lot of innovation. But, I believe I'm excited to be here because I know that the mission is the democratization of proteomics to just get it out there, get it in the hands of the best and the brightest.
All the great analysts out there, right? All of the great big data folks who have developed such great tools in that genetic space. And I'll also say, when you were talking to Chris Whelan well before this whole UKB PPP project came to fruition, there was no guarantee that Olink was going to be the chosen technology. It's such an honor that the tools and the priorities that we've thought were important, specificity, all that, that those were also important and continue to be very important to pharma. I'm going to also just point out that we're now at, just as of this year, at about 5,400 proteins and a really increased, streamlined workflow, increased throughput capability, which is very exciting to see too. Any last Yeah. Any last comments? Yeah. Please go ahead, Evan.
No, I will. And I hope I can say this because, I'm an Olink employee but this is a Proteomics in Proximity podcast, right? So I do think that there's going to be multiple tools eventually that are going to answer these questions, right? I mean, I'm not so myopic as to think that Olink is the only tool out there. I think we have some really compelling attributes for the large scale projects and for these large clinical analyses. But I get excited about continued innovation across, the earlier side of the research spectrum where there could be tools that can rapidly tell you about all these different proteoforms and phosphorylation states. And it's a community, right? That's coming together. Yeah. And I think that, there's so much has happened in the last, decade that I've really been focused in this space, and it's going to continue to evolve. And, I'm grateful that, we've gotten 13 companies together to do something really big. We continue to be integrally involved in the strategy of, drug development from a large number of the world's best companies. And I just think that it's all leading to a more efficient process, right? I mean, I have X number of years on this planet. I want my time to be spent, making a difference for my kids and their kids. And I truly believe that this kind of work is going to enable that. So thank you for having me on.
Our pleasure. Sarantis,
that was great.
Any last words from you?
No, I mean, it was great. I think, Evan, it was great to hear your perspective. And I agree with you. I think the proteomics is going to lead the research from now on. And we're going to see a lot of papers. UK Biobank is only the beginning. And we are looking forward to upcoming projects, actually.
Fantastic. Well, that's it for us today. Again, thank you, Evan, for joining us. I also want to...
Thank you very much.
Yeah, I think there's a couple of authors we may not have said clearly, and that was Fayez Zannad, who was integral in this, and Milton Packer. I don't think we mentioned Milton, who both were integral in really understanding and identifying repurposing opportunities in the Empagliflozin.
Empagliflozin. I think there's in there.
Empagliflozin. Anyway, so I just wanted to click on those and we'll put those into the show notes as well. Thanks as always to my co-host Sarantis.
Thank you, Cindy.
Yeah, of course. If you enjoyed listening to Proteomics in Proximity, please share it with a friend or a colleague who you think might also enjoy it. Maybe we'll get more than 11 listeners. We'll see. And remember, you can reach out to us at Proteomics in Proximity at pip@olink.com. And, any feedback, positive, negative, who should we interview? We would be grateful for the suggestions and the feedback. And with that, we'll close. Thanks, everyone.
Thank you.
Thank you.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.